Yeah,
And thank you very much for giving me an opportunity to present something about Schrodinger suite, which is a really exciting software suite, which has many things to do in the field of computer-aided drug designing. And I hope at the end of this presentation, everybody will get to know about more about Schrodinger suite and what are the things which are present with the Schrodinger software and what are the things which you can do actually. So this is what I would like to say. So before starting the presentation, I would like to tell you one thing that I'm going to cover mostly on the software aspect of Schrodinger suite, including little bit basics of computer aided drug designing and in general drug designing. And finally, I will also touch up upon the case study like how efficiently one can use software to get the best result out of it. And truly said by Mr. Raghu in the short introduction, like there were a few case studies by Schrodinger itself. Even as a software company, we have our own drug discovery group, and that works on some critical problems in drug discovery, and we got success actually. And I will briefly touch upon it, but in detail, like we will see it in upcoming presentations, what it is about, but I will just touch it upon various topics. So I hope at the end you will like it. So before starting the presentation, can anybody please confirm, Sonalika, whether the screen is visible and my voice is clear
Yes, so
enough?
your screen is visible, sir.
Okay, great. So, yep. So I would like to start from instead of just starting the presentation, here is my email ID. So even you can note it down, and in case of any queries or if questions are not addressed here in this particular presentation, or if you want to consult us further, do not hesitate to write me back. So I'll be always happy to help you out and provide the suggestions. So please be in touch and please feel free to write me. So with this, like I will be mostly focusing on how one can transform the drug discovery using computational modeling, and I would say more advanced methods of computational modeling. So most of the time you have heard of molecular docking, but there is a lot more in the field of computer aided drug designing, and this is what we are going to see now. So if you talk a little bit about if you know about drug designing, so or how do like a drug gets developed or how it binds to the receptor. So basically, if you go really into the basics, for every drug, like it must have some target where it gets bind and show some pharmacological action. So this all resembles to the like lock and key model. In very simplistic way, you have a lock, you have a key, and then your key fits into lock. It when you move your key, it induces some changes inside the lock. We call it as a conformational changes, and somehow it changes the internal structure of lock, and your lock gets open or it gets closed. So for that, what is important thing is first of all, like those conformational changes which are happening inside the lock, and the key which by using that thing we can make those conformational changes. And we need to know that thing, like key is very much specific to lock. So not every key fits in every lock. So your key and lock combination is very much unique. And now let's come to the biology. And if you see, like there are many such keys are available in the world, but for your lock, with just a single key which works very fine. So same way, there are many chemicals which are available in the chemical space, and its number is 10 to the power 60. That many huge number of possible chemicals are available, but for the particular receptor or for particular, so in our case, in our body, the receptor is nothing but the protein. It can be DNA, it can be RNA. So it's a biological molecule. So for that particular protein, you have a particular key, and whole idea of drug designing is to find that particular key which fits best in your lock, means that particular protein. So so there are various ways to find out that key which fits particularly inside the lock. But as told by Mr. Raghu, that before it was a serendipity, like people used to try many keys, and out of that, maybe they could find one key which fits better. And such accidental discoveries do happen, just like a blockbuster drug, paracetamol you have. Like it was discovered long time back, and it is still working very fine. But there was no such a hardcore research or computer-aided drug design things were involved in it. But nowadays, like because of emergence of this method, so what do you get because of those emergence of these computers and computer aided drug designing methods is you will get the microscope. What microscope? You will get the molecular microscope, through which basically you can see the internal structure of lock, like how the lock looks like. So same way, if you know the internal structure of locks, you know where are the keys, where are the levers, so you can design your key in a better way. So the same thing is applied to the receptor. You have a protein structure, and if you could see inside that protein structure, what are the amino acid residues present inside it, what are the functional group present inside it, how it looks like, what is shape of it, then definitely you can design a better lock, a better key, or better drug molecule against that protein. So this is what the advantage you get it because of computer aided drug designing methods. So we call it as a molecular microscope because you will be able to see everything at atomic level detail. And consider Schrodinger suit as a molecular microscope. So it allows you to see what is inside the lock, what is inside the protein structure. And with the help of that, we proceed further with the designing of molecules. So even computer-aided drug designing, it's a vast field. There are many things which are involved in it. But overall, when we talk about drug designing, it is broadly classified into structure-based drug designing and ligand-based drug designing. So here, whenever we say structure, so structure resembles to the structure of receptor, in structure of lock, structure of protein. So if you have that information available with you, means the structure of protein, then we can go for structure-based methods like molecular docking, molecular dynamic simulations. But there can be a case when you don't have information of structure of your receptor. In that case, we can also rely on ligand-based methods like pharmacophore, QSAR methods, or shape-based screenings. So we are going to see all those methods. There are many such methods which are available inside these structure-based and ligand-based methods, and we are going to see it in subsequent sessions. But right now, we will mostly try to understand what it happens actually. So yeah, overall, like we mostly deal with the molecular docking process in which you have a receptor structure. So protein looks like something like this. It's a huge structure. It has particular shape. It has particular volume. It has particular binding pocket. And with the help of software, we try to dock the millions of compound inside the protein structure. Millions of compound means millions of keys, and out of those millions of key, with the help of molecular docking, we generate various poses, calculate their energies. We try to identify a suitable key which fits better inside the binding pocket. And then we try to further modify that key using further substitutions on it. So in this way, we try to optimize that lead molecule, and we calculate various parameters like energy, its interaction, and based on all those things, we try to shortlist the best molecule out of it. And this is the molecule we often take it further, and we go for synthesis and we check its biological activity. So this is what the minimum thing which we do in computer aided drug designing. And again, let me tell you, this is the minimum thing which we do, but the sky is again open for you. There are many methods which are available. So before proceeding and for knowing more methods, like it is worth to know the history of it, like from where it started. So maybe many of you are aware of this history, or many of you are unaware of this history. But it is history is always interesting, so it is better to know it. So from where it all got started, so it was started really long time back. If I remember correctly, it's 1960s, when there was a scientist called Martin Karplus. Like he's still alive, the scientist is still alive. And so in 2013, if I'm not wrong, he got the Nobel Prize for his discoveries, and his discovery was developing a force field. So it all started in that time, 1960s. Like during his PhD, he developed force field, and later on he continued with it. So force field was really necessary for studying those molecules or studying the structures as a molecular model. So imagine like in 1960s it started, and then people started working on it mostly in academics, and then it took around 30 years to develop that field and successfully applied into the field of drug designing. So the successful example was first reported by Abbott Laboratories, and it was on HIV protease. So they developed HIV protease inhibitor using this rational drug designing, which is help of computers. So this is the first reported study was in early 1990s. So you can imagine like it took 30 years to develop that field and actually implementing in it. And after that discovery, when that paper got published, everybody came to know like it was a revolution in the field of drug discovery, like because people got something to see inside the protein structure, and that is what your molecular modeling software is. So the thing is, after that, every pharma company, every academic started using all those softwares, started even developing, updating that software, and then rest is the history. So there are many such very well-known drug, like you have often heard of Tamiflu. If you start searching for computer-aided drug design success story, this was one of the starting first example, like who got success, Tamiflu, and there are many such drugs which are successfully developed through computer-aided drug designing. And nowadays, like every single drug which you think of, like it passes through the process of computer-aided drug designing because it is method of virtual screening, the fastest method. So there are many such examples by various companies, and you can find all those things in a literature also. So what is basically the power of all this technique of molecular modeling? So here is the video which depicts the power of it, like up to what extent one can study the biological phenomena or physical system with the help of computers. So you really don't need to look at water in the glass, but you can simply perform molecular dynamic simulations of water at different temperatures. So let me tell you the example. So this is what the first left hand side video you can see. It is basically a molecular dynamic simulation of water. So you have a single water which is present here at the middle red is oxygen, white is hydrogen, and which is surrounded by other water molecules, and we simulated it at a basically room temperature where water stays at liquid state. And you can see these yellow dash lines are nothing but the intermolecular hydrogen bonding which are happening with the surrounding water molecules. And if you can carefully see, you can see at any given point this particular single water molecule is forming around 4 hydrogen bonding interactions with surrounding water. So this is what is the reason why water stays in a liquid state at room temperature. But now let's take another example. I increase the temperature again in the computer. See, virtually I am doing all those things. When we increase the temperature up to the boiling point, and you can see when we simulate thing, you can suddenly see the water molecule and the number of hydrogen bondings are reduced to 2 or 3. So at any given point, it is forming 2 hydrogen bonding interactions, means less number of hydrogen bonding interactions, and that prompt us the water goes into gaseous phase. So this is what well-known phenomena it has. It reduces the intermolecular hydrogen bonding interaction because of increase in temperature. And when it happens, the state of water get changes from liquid to gaseous phase, and you can capture this complete phenomena with the help of computer and the simulation. So you really don't need to look it at water place in your beaker, or you don't need to boil it. You can just do it in a computer. So this is what the importance of computer aided drug designing and field of molecular modeling. So you can all simulate everything in the computer itself. So other than that, like it is worth to know like from where it started. So when all those things started developing, and you can see like industry started using it. So it was in late 90s. Then when two scientists came together, Professor Richard Fishner and Professor William Goddard from Columbia University and California Institute of Technology. They came together. They were really eminent scientists in the field. They are still alive. So they are a founder of Schrodinger's software. So they started basically developing this software. They thought, okay, this is the software which is the future. And then they started working on the field. They started developing the software, each module one by one. So looking with the mission, with the single mission, of course, to improve the human health and quality of life by revolutionizing this particular drug discovery. And not only drug discovery, but one can use or you can study various materials also with the help of computational methods. So this is what their aim was, and they started implementing all those things in the software, and they started growing it one by one. And now after like 30 years, this is the complete software suite which we have, and which has number of modules present and across the globe. Like as a Schrodinger company has grown that much that across the globe we have presence at various places. Mostly our R&D team lies in eastern and western coast of USA, but other than that in UK, Germany, India, and Japan also we have office where we do interact with the scientists who are using Schrodinger software. So, and then what comes with the Schrodinger suite is there are various type of small subsuits which are a part of complete Schrodinger suite. So it can start with the simple way of visualizing molecule in the pi mole, or if you are working in case of DNAs, proteins, RNAs, antibodies, antigens. So we have a tool called Biologic suite where all modules come together to help you to study all biological molecules. And if you are interested in drug discovery, means small molecule drug discovery, so we have a special suite for that, which has all compilation of all modules which are necessary for drug designing. And not only that, even for materials, if you want to study materials like batteries, nanoparticles, carbon nanotubes, so all those things one can study using material science suite. You'll be surprised to know that even ISRO can use material science suite. They are also using it, not only ISRO but Boeing also. So in this way, like it has a wide usage, and not only that, even Discovery Informatics is the small subsuite package which we have, which helps you to analyze the huge amount of data which is generated by day-to-day exercise in the field of chemoinformatics. So this is all come together with the Schrodinger suite complete package. So let me introduce those package in particular way. Like in case of small molecule drug discovery suite, which helps you to assist with particularly drug discovery process or drug discovery methods. So we have various tools, like for example, which I have shown, which is most widely used is protein ligand docking, in which one can perform molecular docking of huge library of compounds in fastest way inside the protein structure. Not only that, but in case of general molecular docking or traditional molecular docking, we consider protein as a rigid molecule. But with the help of another module called induced fit docking, which is again part of Schrodinger suite, we can also consider the binding pocket amino acid residues of protein as a flexible, so that it helps you to understand the protein structure and its conformational changes in better way. Not only that, there are various ligand-based methods, as I said, like pharmacophore-based methods or shape screening, in which one can use this method to screen the like compounds. So, for example, you have active molecule, for example, paracetamol. So, if you want to look something which looks similar to paracetamol, you can use pharmacophore or shape screening to look for similar type of molecule from the whole database of billions of compounds. And similarly, there is a method called 3D QSAR, through which one can generate various contours and understand the chemistry of that particular molecule. And accordingly, you design the number of compounds. So there can be a case when we don't have like information about the binding site which is in the protein structure. So we have a tool to analyze that particular binding pocket using sitemap. So sitemap is the name of tool which helps you to see what is basically present inside the protein structure or inside the binding pocket. So this is what I was talking about. So if you know what is inside the lock, so this is what the tool which helps you to see what are the various properties inside the lock and inside the protein structure. And once you get that information, one can of course use it for the drug designing. Then next thing comes into like core hopping. So core hopping or scaffold hopping, if you have heard of. So this is the method which is very well known, used in the field of drug discovery by industries to generate a new structure of molecules to skip the existing patterns even. So you can basically look for various type of cores which are not existing in any reported patent, and in this way they can get to know about novel molecules and they can patent it, and of course like they can earn a lot of money out of it. So you can also perform various type of energy analysis on the proteins or protein ligand complexes to understand it better, like which functional group of protein is more important for binding to the receptor energetically, how favorable it is, and that can be done. Other than that, basically there is one revolutionary tool called library enumeration. So you can see like you have a ligand which has some basic structure which is constant, and one can generate basically millions of compounds just by single click and performing various substitution at the given point. So this is called as combinatorial chemistry. Like you don't need to draw each structure one by one and generate a lakhs of compound. No, that is not the smart way of doing it. The smart way of doing it is enumeration, automatic enumeration. You just need to tell the software, okay, I want substitution at this particular position, and software will do all type of possible substitution in it. So this is what is present with the Schrodinger suite, and I must say it is very much revolutionary in nature. Other than that, if you go for advanced level of molecular modeling, and so you have often heard of the term molecular dynamic simulations, in which here we talk about actually simulating the biological system in biological environment, how it looks like if you increase the temperature, how it is actually present inside the cell, inside the body. So we call it as a molecular dynamic simulation, and we can simulate it in a real time to see what are the conformational changes which are occurring in the protein. Means I was talking about the structure of lock. So you can see like what are the conformational changes which are occurring in the lock. But here the lock is moving, and you need to know like how it is moving. So how conformational changes are happening, how it affects the ligand binding. And not only that, you have a very another unique tool which is called for covalent type of docking. So covalent docking is nowadays very much in the talk and very much popular, because in case of COVID research, there is a protein called mpro, and it is reported with the covalent binder. So that is why it is in very well known field now. So for to study this covalent binding and the phenomena, you can also study it using the Schrodinger suite. We have module to do that. And of course, if you go for higher level of studies, like you can of course refine the X-ray structure based on the electron densities and based on the energetics of all those residues. So for that also we have a module. You really don't need to rely everything on the experiments. Like we can also do these things using software and Schrodinger suite. So if you go for higher level of molecular modeling studies, the quantum mechanics is the often heard term. So we can also do the quantum mechanical calculations in Schrodinger suite to understand the structure and chemistry of molecule in better way. Then comes to the really advanced modules, and I must say this is a very popular module in industry because it is giving revolutionary result. And the module name is water map. Even if you can see here, so this is basically a tool which helps you to understand the thermodynamics of water. So thermodynamics of water, it is very the term which we heard in plus two, our plus two. It's a simple thermodynamics like enthalpy, entropy changes, and we can of course study those enthalpic changes whichever are happening in water and when those are present inside the binding pocket. And based on that, you will come to know about the very particular thing in biology, and that particular thing is whether your molecule water is happy or unhappy when it is present inside the binding pocket. So simplest thing is, you can see here there is a protein surface. It's all many water molecules which are highlighted in sphere, and those are present inside the binding pocket of the protein. And you can see there are a few spheres which are water molecule are in red color and are in green color. So green color is like a happy water molecule and red color is unhappy water molecule. So once you identify which one, which candidate is happy, which one is unhappy, so the simple logic, it is always easy to replace unhappy one, and it is difficult to replace happy one. So overall idea is to replace the unhappy one to design drug in better way. So this is what which helps us to understand the water thermodynamics and drug designing in better way. Then here it comes the another module called free energy perturbation FEP plus, through which basically one can predict the binding energy of ligand molecule inside the protein structure with the accuracy of zero point 0.6 kilocalorie per mole, and it's very rare, and I must say it is very much revolutionary module which was by Schrodinger. And you can see here in this particular example, like with the help of software they could replace basically during the go, like they could replace the they could adjust the another functional group inside the ligand, and because of that functional group you can see it has changed to the halogen chloride. Because of that chloride, how much the binding energy got changed, and we are going to see all those things in more detail in coming presentation, so don't worry about it at this point. Just remember there are various things which you can do it with the Schrodinger suit. This is what is the another example in live molecular dynamic simulations, like it tries to modify the molecule inside the binding pocket and try to compute its binding energy. So this is what it comes with the small molecule drug discovery. So it was all about studying small molecules or doing the small molecule drug discovery. But other module which I was talking about is the biologic suite. Means whenever you are working on proteins, antibodies, DNA, RNA, antigens. So if you are studying those kind of things, we often call it as a biologics modeling. So we can do it with Schrodinger suit. For example, you can design antibodies, you can humanize antibodies, you can study antigen antibody interaction, you can develop a model of protein using homology modeling. You can study various phenomena like protein aggregations and what are the properties of it. Not only that, you can also study how protein binds together, how protein DNA interacts, how protein RNA interacts. Those kind of things you can study with the biologic suit. And not only that, even in case of if you want to modify the protein, it comes under the protein engineering. For example, residue scanning like alanine scanning, cysteine scanning, disulfide prediction, identification of reactive residues to engineer your protein in better way to improve its activity. So you can basically make a thermostable protein for your protein engineering stuff to increase its catalysis or to increase its stability. So we can have those applications also present in the biologic suite. And when I said like discovery informatics, so informatics, whenever you work on various projects in the field of computer aided drug designing, in chemoinformatics, you come up with the huge amount of data. And it is not only about generating data, but it is the discovery is all about analyzing that data and get the information out of it. So this is what the discovery informatic tools which help you to analyze the data in real time. Not only analyzing the data, but consider working live project which is going on and 10 people are working on single project at same time, and everybody is sitting across the globe, and still they can work it in a real time all together. They can do all type of modifications at given point and everybody will else will come to know about it. So this is what the type of collaborations which we could make. We call it as a uh, basically project management thing or enterprise discovery enterprise. So means everybody else come together and they start working on the same project, and this is what very much common in the pharma industries, and not only industries but even in academics you can have live collaborations where all scientists come together and they work on the same project at same time, and they can get more information out of it. So this is the way how discovery informatics helps. And not only that, in discovery informatics is all about analyzing data, as I said. So you can do various type of clustering to analyze the things. You will have like huge amount of data. When I say huge amount of data for the chemoinformatics way, means you will have huge descriptors, huge amount of descriptors for many billions of molecules. It finally ends up with the gigabytes of data, and you need a very powerful tool to analyze that data in various ways. So you can search it based on clustering, based on its fingerprint matching, based on its descriptors. You can build various type of models. We call it as a QSR models, and all those things comes under this discovery informatics to help you to better understand the things. Then things comes to the finally visualization. So visualization means nothing but the representation of data. So at the end you do your research project, you come up with a good results and everything, but it is always necessary and very important thing is to present that data in the nice way. So to do that thing, to present the data in very nice way, we have a very powerful visualization tool called PyMol. And with the help of that PyMol tool, like you can animate the thing, you can present it in the best way so that it is understandable to the researchers. And this is also one of the key point to get your data published in good journals or to get the recognition by getting on the cover page of the journal. So this is what the live example we have for the visualization or the data representation. And then in brief about material science, so what you can do with the materials and material science. So, you know, like whatever is existing in the world, whatever you can see is all materials. It all considered in the material. And of course, we can consider, we can study all those materials like carbon nanotubes or nanoparticles or any type of material, even batteries. You can study it with the Schrodinger suite. So what things you can study it with the Schrodinger suite is you can study its energetic, you can build various type of complexes like metal complexes, which has very weird kind of geometries. And you can screen basically various type of structures which helps you to understand it in better way. For example, here is a virtual screening of battery additives. Means with the help of this software, you can find out which additive works better in your battery. And not only battery, like we can apply these things also in case of designing the better material for rocket, which sustain in the space at particular temperature, particular pressure, and all those things. So you can look for various type of unexplored type of molecules as your material for your particularly for your particular study. So for example, here is an example of a film deposition on the top of some layer. So you can also study that. So this is what it comes under all molecular modeling. So there is a often question people do ask, like if I can do the things with molecular docking itself, then why do I need that many tools? So far, if you remember, like I have presented like more than 30 40 tools. So there are many such tools which are necessary, and why do we need those tools? And basically, who are using those tools? Is it really worth to look for that many tools? So my simple answer will be yes, you need that many tools because the thing is very much complicated. So if you see in the field of drug discovery, the overall process itself is very much complicated and lengthy. So if you have heard of the process of drug discovery from target identification up to the drug in market, it takes around like 13 to 15 years of time, and that's really huge time. In that like 13 to 15 years, like generation get changed, and you really don't want to wait for that much long time, but we need to wait. That's the critical thing. We need to spend that much time on drug discovery phases. And it's not only about time, but it is also about the money. And so in our world, like time is always money. So as you spend more time, it comes with the amount of salary for researchers. Many researchers are involved in it, their time, their money. So everything comes into picture, and it all end up with around 2 billion US dollar industry for each drug molecule. And if you calculate it in Indian rupees, it is almost 18,000 crore of rupees. It's a huge amount of money. So, but if you see it critically, there are various phases of drug discovery. If you think smartly, like where you can work on, so you can see there are blue color regions and orange color regions. So here is the orange color region, which is a more time consuming step for each step. We call it as a clinical phase, means preclinical, phase 1, phase 2, phase 3. And basically, as a researcher, you can't really do anything much in this clinical phase, because whenever a molecule gets put into clinical phase, it's a single drug molecule which we administer in the patients and study its properties. But we can't really modify that molecule. We can't play with that molecule, because what we can do as a researcher is what clinical scientists do. They observe the properties, they observe what effect that particular molecule is giving us in the body. That's it. Means they can only observe the thing, but they can't play with the molecule. But if you see smartly, there is an initial phase of drug discovery which spans for around 6 to 7 years of time. out of 13 to 15 years. So you can see like it all comes up with the initial phases where you perform high throughput virtual screening, you identify the target, you identify the molecule, you optimize that molecule. In short, like you can play with the particular molecule, you can optimize it, you can perform various substitution, check its properties. And once it is all done, you end up with the one molecule, you put it into clinical trial and that's it. Afterwards, like human can only analyze all, only look for the things which a molecule does with our body. But the researchers can play initial its role in initial phase of drug discovery. So with the help of computers, like we can work on that initial phase at various steps, and we try to reduce the number of animal studies. We try to play with as many as possible number of molecules to identify the best molecule out of it. And to work on this particular initial phase of drug discovery, here the software comes into picture, and here that many tools comes into picture. Like with the help of that many tools, you can study all type of millions of compounds thoroughly to get the best result. So this is what the necessary thing which we need to do to reduce the time scale of drug discovery to give the better drug as an output. And why it is like as such it is very much complicated, as I said. And why do you need that many tools? Because it's very much complicated, and to understand its complexity you need to understand the thing. It is really complicated. So here is the example where you can have various drug molecules, and there are various number of properties which you need to consider whenever you design a drug molecule. Like it should have good binding, it should have good potency. Not only that, it should have good pharmacokinetic properties, it should have good metabolic profile, it should have good solubility, and many other things. It should not have any toxicity. So there are many aspects, and I would say there are hundreds of such aspects which you need to consider while designing a molecule. And the overall drug discovery is identifying about the star molecule which comes under all aspects, and it should satisfy all those criteria. So finding this star molecule is really difficult, and even I would say it is harder than the rocket science, because rocket science also follows the laws of physics, and if you project the rocket, it may go in the right direction. But if you consider the same thing in case of biological system, when you put drug molecule inside the body, you never know how it is going to be affected. So we can't have any control on it. Just like once you take the tablet, you don't have any control on it, like where it goes, where it binds, what effect it will show. It can show any type of effect. And with the help of computers, the drug discovery scientists, basically our duty is to manage all those properties in this particular star molecule. That is the reason. I hope you got the complexity of it. So the thing is, one can get through all those things with the simple logic that if you test more number of compounds, the chances is better to get the good molecule as output. And this good number thing, like 10-20 years before that, people used to synthesize like 2000 molecules, because that was the capacity of any synthetic chemist. And out of which, like they could end up with the one molecule in clinical trials, and eventually if that get fails, the whole project get down. But now with the help of advanced computational modeling, we have softwares, we can basically screen not only thousands but billions of such compounds. I would say 100 billions of compounds. You can screen it with the help of computers rapidly, fast, and with accurate predictions. And when we apply this all computational filter in it, the chances of getting a good molecule out of it is huge. And again, like once you get that molecule as an output with that huge chances, the chances of failure becomes less. But still, but the still, like you, whenever you put molecules in clinical trial, you never know like whether it will pass or fail. But we can predict all those properties at early stage of drug discovery to reduce that loss. So this is what the important thing which we need to always consider as a drug discovery scientist. And here I would just like to give you a brief introduction on, so I was talking about all those things, how this software helps, and one should go with, what are the things one should go with, and what are the ideal things we should consider. But this is what was actually happened. So in Schrodinger, we work on a project called acetyl coenzyme A carboxylase inhibitor design, along with the Nimbus Therapeutics, which is again contract research organization. So Schrodinger and Nimbus get collaborated, and they started working on the project called ACC. And this is what the project was mentioned by Mr. Raghu in initial presentation. So they got the success by using all type of computational methods, and they got the molecule with IND approval within a span of 16 months. If you see this line on bottom, so it's a time scale, like they generated various type of molecules. Initially they used computational screening, not only computational but experimental screening on selected compound. They optimized the hit molecule, and finally they got the lead molecule and they filed for IND approval. And whole process took only 16 months of time, and this is not even 1.5 years of time, you can see. And initially we were talking about that initial phase of drug discovery was for 6 to 7 years, and now we have reduced with the help of computational tools to just 16 months, and that is called as acceleration of drug discovery. That is called as saving of money, not only money, saving of time. So in this way, we can accelerate the process, and the best example is in front of you, like COVID drugs we have. So this is what is the example of accelerated drug discovery. They could screen the various molecules using drug repurposing, and you got remdesivir as an output against the COVID, and this is what people are using. So it all happens because of computational power, and not only computational power, but the scientists from various disciplines, when they all come together and work rationally. Rational is the important word here. If they work rationally with the help of all tools, they could get success within a stipulated time. And this was the breakthrough. Like when Schrodinger got success with this particular molecule within a span of 16 months, they sold this complete project to Gilead Pharma, and it was for 1.2 billion. They made huge money out of it. So they showed to the drug discovery industry, like this is how one could efficiently use the software to develop a drug against target. So this was the successful example, and this successful example will be again explained in detail in upcoming presentations. So don't worry about it. Just keep in mind that we have a success story. And right now, this particular molecule is in phase 2 clinical trial, and it is along with the Nimbus Therapeutics. So in this way, like with Schrodinger, there are many such projects which are going on, and many such projects are in advanced stage. And not only with that, even with Indian pharma industry, this is the Spark. Spark is the subsidiary of Sun Pharma, which is the Indian pharma industry. Like even we collaborated with that.